we're Rage Against the Phage, and we're here to teach you a thing or two about viruses. To start off, what are phage? Phage are a special type of virus that infect bacteria, and they're super important. It's said there are more phage on the planet than there are any other organism combined, including bacteria. Phage are also important for their role in nutrient cycling in ecological systems and for their role in bacterial evolution. Our phage was collected from a garden in Venice, California. The soil it was collected from was dark and damp. In order to isolate our phage, we added it to a pre-made enrichment broth and filtered it through a filter small enough to not let any bacteria through. This was then added to a sample of Mycobacterium smegmatis, or M-smeg, and plaques were seen where the phage had killed the bacteria. Phage can enter two main life cycles, one of which is lytic. In the lytic life cycle, the phage attaches to a bacterial cell, inserts its genome, which is quickly replicated, and the phage is assembled. Then the phage lyses the cell and releases more phage to repeat the process. The other life cycle is lysogenic, where a phage attaches, inserts its DNA, which circularizes and becomes part of the host DNA. This is called a prophage, which we will go over later. This way, when the bacterial cell divides, it also replicates the phage DNA. Some phage are temperate, allowing them to enter both life cycles, lytic to lyse the cell and release more viruses, or lysogenic to become part of the cell's genes and replicate with the cell. Our phage is a temperate phage. Life cycle can be determined by looking at the plaques a phage makes on bacteria. If it's lytic, it'll make a clear plaque. Lysogenic will make a halo or turbid plaque. Our plaque was temperate and made a bullseye plaque. Now we'll look at the way phage communicate. We'll start by looking at bacterial communication, or quorum sensing. Bacterial cells can release small molecules to communicate with surrounding bacteria to help form colonies. More of these molecules cause different changes in bacterial populations. So the question is, do phage do this? And, excitingly, it looks like they do. A recent study found that some Bacillus bacteriophage may secrete a quorum-sensing molecule, allowing them to communicate with each other to begin the lysogenic life cycle. So, does our phage do this? In order to find out, we created two types of media, one with just bacteria and one with bacteria and phage. These were allowed to grow, and then we centrifuged them and collect the liquid at the top. These were then centrifuged through a tiny filter that the phage couldn't even pass through, and both of the medias were recollected. These were then added to M-smeg and phage and grown on plates to find colonies and plaques. We expect to see more lysogeny in the conditioned media and more lysis in the controlled media. Remember prophage from before? When a phage inserts its own DNA into the bacterial DNA? Well, as it turns out, there are a couple ways you can make a phage switch from a lysogenic cycle to a lytic cycle, including UV radiation, antibiotics, and quorum sensing molecules. We chose to do an experiment with UV radiation. We started by making a liquid lysogen culture. A lysogen is a bacteria with a phage genome inside. We did this by picking a bullseye plaque, streaking it on a plate to purify it, and picking an individual colony to grow in a liquid medium. We then ran induction in their liquid forms and separately on the plates. The liquid lysogens were put on an empty petri dish, exposed to the UV radiation, grown, centrifuged, had the phage collected off the top, and then added to M-smeg for a plaque assay. The plated lysogens had the plaque assay done first, were then exposed to UV, allowed to grow, and the morphologies were observed. Because we're inducing the lytic cycle, we hope to see more cell death through clearer plaques compared to plates not grown in UV. Looking ahead, we're going to have to do some genomic analysis to find out which genes are related to the life cycle and which genes make our phage a temperate phage. So what does this mean? This means we have a better understanding of phage life cycles and how we can change them, and a better understanding of how they fit into our lives. We hope you enjoyed this video and maybe even learned something along the way. Thanks for watching.